Kate Scow here. This is Soil Microbiology, and this is our last lecture for this course. It's actually called Lecture 9, even though we've had Lecture 10 already. I reversed the last two lectures. So Lecture 9 is about microbial transformation of this enormous variety of other elements that we haven't considered at all. We've had a strong focus on carbon and nitrogen, and then we talked about pollutants, a variety of different organic compounds, but there's a whole lot of other stuff going on out there, and I have on this cover slide of the lecture this variety of different elements that microbes also have interactions with in different ways. And there's some very cool pictures that show an iron oxidizing bacterium who has converted ferrous soluble iron to ferric iron that you can see precipitating out around the bacterium. And also Schuanella, who is adorably, as this picture says, spinning nanotubes out of arsenic and thiosulfate. Schuanella can use sulfur and creating these really complex, interesting structures in the environment, which is what microbes do, and we see evidence of their actions all around us, particularly at microscales. Okay, so let's just back up and talk very generally about all the different kinds of reactions that microbes are involved with with minerals. And some of these types of reactions you've seen before because they also occur with nitrogen and carbon. So let's just rattle through these to get the big picture, and so that you know that these same kinds of processes are involved in a variety of different elements, particularly mineralization and immobilization. There you see it again, because life forms are made up of all these different ions that are used as nutrients, and whenever something dies and is ready to enter the universe, microbes are going to break it down and convert to mineral the forms of convert to minerals all these ions that are existing in organic form, but now will be liberated. And vice versa, microbes need a lot of those elements as nutrients themselves, and they are going to immobilize or assimilate them. And this will also depend on a carbon to whatever the element ratio that's going to determine whether it's net mineralization or immobilization. But for many of these other ions, there's so small amount needed in the cell. So those carbon to X ratios are going to be super, super, super high and often not an issue because they are in such low concentrations. All right, these ions can also be oxidized if they're in a reduced form, and they can serve as electron donors. But also they can be reduced if they're in an oxidized form, serving as an electron acceptor, or they may just reduce the elements without getting anything from it. There are these examples of these kind of accidental reactions going on all the time. Either that or we just don't understand it, so we call them accidents. That'd be a very human thing to do. Okay, complexation, so that's chemical reaction between the ions and some microbial produced organic compound, a ligand, like a chelate, that might be used to access iron. We'll talk about that, or a methyl group, a methylated form of the ion. Then we can have indirect transformations of elements from all the different things that microbes do, changing the pH, the redox, that can result in mobilization of ions that might have otherwise stayed put, or solubilization. So these are super important in soil, is all the ways microbes shape the environment, causing the elements to behave differently. Toxicity could be another issue. Some of the heavy metals like aluminum and cadmium and chromium are super, super toxic. And particularly under some conditions, way more than like iron, nickel, these other elements. But the bioavailability, the bioavailability is going to be an issue where if a soil has a lot of organic matter or clay, perhaps those heavy metals will not be very available and will not end up actually being that toxic to microbes because they can't get access to them. A lot of elements have very low solubilities. So in a lot of cases, particularly like cations, they're going to be not very available and are often going to be limited. So a couple of interesting things are eukaryotes have their electron transport chain internally in their X bacteria, those mitochondria in them. So that limits some of the kinds of reactions that they can carry on with elements that are out in the environment because they cannot have physical contact between their electron transport chain and these maybe insoluble precipitated minerals. In contrast, prokaryotes, because their electron transport chain's in the plasma membrane, they can actually colonize close to what would be insoluble substrates like elemental sulfur, iron sulfides, iron oxide, manganese oxide, and then they can convert it, for example, with these oxides, reduce it, or if it's a reduced compound, oxidize it, even though they don't take up the ions directly into their cytoplasm. But through this physical contact, they can actually shuttle some of the electrons back and forth between the compound that they're using as a donor acceptor and their electron transport chains. It's super cool. Okay. I just want to mention that remediation, just in general, when we're talking about these other elements that are not organic compounds, they don't go away. They don't degrade and disappear. They're always going to be around. So the best that you can do is accumulate them, maybe into particular areas, and then perhaps harvest them then, or gather them up and dispose of them. Or you could use surfactants like we talked about a little bit in the biodegradation lecture, and that might be able to wash away the elements from like a contaminated soil, as long as you have a place to collect them in. And then also phytoremediation, which is plants engaged in some way in remediation of contaminants. That can lead to plants accumulating metals. There's some plants that are really great accumulators, like of nickel or other heavy metals. And then when they accumulate them, then the plants are harvested and then, of course, have to be disposed of as well. But you could use this to kind of mine those toxic chemicals out of the soil, or if you wanted to harvest them, if they were gold or something that you valued, you might be able to use plants to help gather them up. And microbes do play a role in the rhizosphere of those metal accumulating plants. Okay, let's start talking about some particular kinds of elements that are really important for different reasons. And this little table up here is a really good one because it guides you through some of these major elements that you're likely to find in soil. And this has in columns the kinds of interactions that can occur. So we have mineralization and mobilization, oxidation reduction reactions, or methylation, which is that conjugation, one of the kinds of conjugation reactions. So we can look at these groups like phosphorus, potassium, calcium, magnesium, copper, and zinc, all under oxidation reduction reactions, and they're not methylated. Iron and manganese, which we already know about from because we know they're electron donors and acceptors. Yes, they do engage in oxidation reduction reactions. They don't methylate, but of course they're mineralized and mobilized. Everything is mineralized and mobilized. Whereas arsenic, mercury, and selenium have the interesting talent to be methylated. They're also mineralized and mobilized and oxidation, go through oxidation reduction, but they are susceptible to this methylation process that microbes stick a methyl group on the molecule for reasons that we don't entirely understand. So that's sort of a general roadmap of these different elements. Let's talk about phosphorus. So phosphorus is, some people think, magnificently boring, and it's largely because it does not undergo redox reactions in nature. It primarily exists at one redox state, at one oxidation state, which is the +5 phosphate. It doesn't change very much. Under sort of rare conditions will it be converted to another oxidation state, but it mainly hangs out as just +5. And so the big issues of phosphorus are it's a major nutrient after nitrogen. It's next in terms of organisms needing it. And it also has huge issues of bioavailability. Phosphorus is not easily available in soil. So when we look at kind of phosphoric dynamics in soil, we really aren't looking at it cycling between different forms of phosphate. We're mainly looking at organic phosphorus mineralizing to phosphate in solution or being taken up. And then phosphate is just game for all kinds of interactions with different components of the soil. So there could be sorption, desorption of it with mineral surfaces. There can be precipitation dissolution, which happens a whole lot, where it's going to form secondary minerals with like calcium and iron and aluminum, creating these really, really unavailable forms of phosphate that may over time be dissolved and become available. And then we have weathering, the dissolution of primary, and that's another way that phosphate's going to be entering the system. So the phosphorus cycle is quite a bit different, for example, than the nitrogen cycle. So +5 state, it's either in organic or inorganic forms. The inorganic is usually with aluminum, iron, or calcium, depending on the pH. There's no gas phase. It's not very soluble. Microbes play a big role in making making phosphate available. They can help increase its availability by producing acids. I think you remember that ways that microbes produce acids are through fermentation, making organic acids, from respiration, making carbonic acid, from nitrification, producing nitric acid, and from sulfur oxidation, producing sulfuric acid. So all of these acids can lead to increased solubilization of inorganic P. That means pulling the inorganic phosphorus out of these kinds of secondary minerals. Okay, and also chelation. So chelation indirectly releases phosphate because it may form a complex with the other ion that is in association with the phosphate. In particular, like iron. The chelate will pick up the iron, and then the phosphate will be abandoned and then become available. All right, immobilization, mineralization. The carbon to phosphorus ratio is around 200 to 300, so way higher than nitrogen. This is the ratio at which you're going to have either net immobilization or net mineralization of phosphate. So takes a lot more carbon to lead to this kind of immobilization. So a lot of organisms have enzymes like phosphatases and phytases, particularly mycorrhizal fungi, that help mineralize that organic form of compound that has phosphorus in it into phosphate. Also, rock phosphate and manure have been used for many years as sources of fertilizer. Rock that needs to be mineralized to become available, manure that needs to be mineralized to become available also, but two different kinds of mineralization. Ecto and endomycorrhizae are really important in accessing phosphate and helping it back to the plant. And I just think it's interesting. I had a colleague when I first came, the guy who made the first mass spec ever and was sort of one of the fathers of biogeochemistry, Connie Dalwich. He used to say that he thinks that ghosts in graveyards were actually phosphine gas. When corpses decompose, they create very anaerobic sites. He thought that under those conditions, the phosphate could actually be reduced to phosphine, to a -3 form. And it's extremely unstable and won't last long, even if it is made, and ignites with oxygen and creates these like ghostly green diffuse gases around them that Connie Delwich used to think, those are the ghosts that people see around graveyards. It's just this phosphine acting up. Who knows if it's true? Okay, an interesting thing about phosphorus, of course, this isn't quite as simple as people used to think of it, but there is this concept of peak phosphorus. It's important to note. Phosphorus is a compound that is mined. It's a resource that's mined from deposits in the world that have high amounts of phosphorus. Morocco, in particular, has a lot of it, 32% of it, whereas U.S. sources are being slowly depleted, although I think there's recently been found some additional ones. But ultimately, the world is going to run out of phosphorus. We don't know exactly when this is, 300, 400 years. But here's the pattern in blue, production of phosphate over time. And the red is extrapolating what they're thinking. So is there a role for soil biology in all this? Probably. It first relies on us humans to be able to take the resources that are now considered waste products where the phosphorus accumulates. Really good sources of organic phosphorus include animal manures, food wastes, and of course our own manure, human manure that's in the form of biosolids, which all could be really good sources of phosphorus fertilizer if there was a way that we could safely apply them. Some of them already are safely applied to agriculture, and then we would rely on microorganisms to mineralize those organic forms of phosphorus, and it would help us deal with this issue of running out of places where we can mine the phosphorus and instead start recycling the phosphorus that we've already mined. A lot of the phosphorus that's used in agriculture ends up being dumped into oceans because of runoff. And that doesn't make a lot of sense, does it? Okay, with phosphorus, sulfur is the next element that we're going to talk about. And its issue is that it undergoes a variety of oxidation reduction reactions, kind of like nitrogen. In fact, it behaves a lot like nitrogen. And this shows you some different sulfur organisms and an area where a lot of sulfur is being produced from a crack in the earth. So the sulfur cycle is really interesting. Doesn't it look a lot like the nitrogen cycle? There's the nitrate, it's sulfate. There's elemental sulfur, the reduced form of sulfide, soil organic matter, and then interaction of the sulfur with the particles in the soil and minerals in the soil that can reduce its availability. So we have mineralization and mobilization, oxidation reduction reactions between all these different forms of sulfur. And then we have an atmospheric phase as well, although nothing quite like nitrogen in terms of the relative proportions. We don't know a lot about how sulfur acts in soil. It's not received a lot of study, and so there's not a whole lot to say about that. But it's definitely coming into the soil in some forms of fertilizer, and also deposition from the atmosphere is causing a lot of sulfur to enter into soils. So sulfur has many oxidation states, and this figure here shows you that it ranges from -2, which are the most reduced forms of sulfur, which are in these sulfide forms, then elemental, which is the 0 oxidation state, then to the thiosulfates that are in the range of -2 to +6 over a variety of different states, to sulfites, which is +4, to sulfates, which is +6. And there can be oxidation reduction reactions along the whole way within some organisms, or just between some of these forms for other organisms. There's a variety of different kinds of diversity of organisms that engage in either sulfur oxidation going this way, boop, boop, or going that way, or reduction of sulfur going in the other direction. And here's some pictures of some of the sulfur organisms, and they often have these black occlusions in them that are sulfur particles, elemental sulfur particles. So you can sometimes see it under the microscope and be able to define these sulfur organisms because of that. So a lot of these are 2 electron shifts, at least on the outer sides, and the organisms get about 50 to 60 kilocals per mole. I think I'm tired. Per mole per 2 electrons transferred. So they get a decent amount of energy from these reactions. A lot of different kinds of sulfur utilizing organisms, and this table just groups them by whether or not they're organoheterotrophs that reduce sulfur compounds, or that oxidize sulfur compounds. So the desulfose, the desulfo is often this root that distinguishes that these are organisms that reduce sulfur compounds. And then the thio root is often used to describe organisms that oxidize sulfur. Those oxidizers are either obligate or facultative lithoautotrophs that are using the sulfur as a form of energy. And they can use either the most reduced form, but they can also use sulfur all along the way that as an electron donor. And obviously if they're starting over here and they go all the way to sulfate, they're going to make a whole lot more energy out of that series of reactions than if, for example, they start with just sulfite as the donor, and there's only going to be a 2 electron transfer there. Whereas here there's going to be from a -2 to a +6. So that's 4 2 electron transfers, so 4 times this. So they're obviously going to make a lot more energy if they're starting here than if they're starting there. Okay, and then there's a lot of organisms, and this is just pretty much everybody that mineralizes and assimilates sulfur. Okay, the really interesting thing about sulfur that we also touched on when we were talking about flooded soils is the propensity of the reduced form of sulfur to form secondary minerals with a variety of cations. And this is a rather complicated figure that is describing that, but essentially showing that when you have H S, the reduced form of sulfur, the sulfides forming, then they're going to interact with any variety of metals that are present, iron, manganese, zinc, lead, copper, whatever is there, and precipitate out as sulfides and accumulate over time. Okay, iron. Iron, the issues there are iron is very unavailable but very needed in lots of reactions. And there we have the siderophores, which we'll talk about in a minute. And also another issue with iron is acid mine drainage. So the iron cycle is not that complex. It's mainly iron is in two forms in nature, in the +3 form, the oxidized form, ferric iron, or in the reduced form, which is ferrous iron, the +2. Iron in the +3 form is not very available. It's very low in solubility and not easy for organisms to get access to. But in the +2 form, in the reduced form, it's very available, it's very mobile, it's very easy for organisms to get access to them. So we have this conversion between the oxidized and the reduced form, and we already know quite well that ferric iron is an electron acceptor, so it's reduced down to ferrous, and then ferrous iron is an electron donor, and it can be oxidized to form ferric iron. But then there's a whole lot of shenanigans going on with other kinds of minerals in the soil that's going to then also influence the access to those different forms of iron. And I think I talked before about the fact that organisms that are prokaryotes that have their electron transport chain in their outer membrane, they can access, for example, that very low solubility ferric iron by actually attaching to the surfaces of minerals with ferric iron, and can essentially either transfer their electrons directly to it, or maybe access anything that just comes off into solution. They can grab it right away. What's interesting is a couple of organisms also produce chelates that are involved in electron transport chain reactions. So chelates, siderophores, right? Siderophores is the specific name for these chelates that bacteria produce, which can form, it's a special kind of organic molecule with a structure that forms complexes with trivalent cations. And often they seem to be organic compounds that microbes produce, in particular to go after iron, because iron is so unavailable and so needed in the environment. And these chelates are quite good at being able to capture the iron at surfaces and keep it in solution until it can get over to a microbe, where they can then pluck off the iron and actually recycle that siderophore back to go get more iron. And this is shown here. Here's this microbe. It produces big, in this case, complex siderophore molecule that has this ability to, like, through its conformation, to form a bond with the iron in these three different connections. And then, so after it picks up the iron, it goes back to the cell, where the iron is transported into the cell, but the siderophore is just sent back out to get more iron. Pretty interesting kind of capability, and organisms that have siderophores are much more competitive in iron poor environments, even though it's expensive to make those siderophores. It's a good investment in environments where there's very little iron, and such environments include like rhizospheres, where there's quite a bit of a battle with organisms competing for iron. But also in the human body, and some of our scariest pathogens are organisms that produce siderophores that are able to wrestle some of the iron in our bodies, which a lot of them are in hemoglobin, and so therefore not very available. Like there's just not much access to iron in the body because they're in these forms that aren't available. So it's hard for pathogens to survive because the body's very iron poor in terms of the availability of the iron. But those with siderophores are really good at getting the iron. And the spooky thing is the siderophores are often fluorescent molecules. And so we have these fluorescent pseudomonads, which are really, really scary pathogens like that colonize burn patients and people who are having other kinds of situations that make them really vulnerable. And they can really wreak havoc in your body. And you can see them by putting them under a black light, and you can see them fluoresce, and you know that that's what you have. And the fluorescence comes from the siderophores. Okay, so a really important concept is that the oxidation of iron is a function of pH. Now, do you remember this from the electron tower? Right? Remember that long ago? That there's two places that iron falls out in the electron tower, and one is just above oxygen and one's a little bit higher up, and where it falls is a function of the pH. So in general, the ferrous iron, the reduced form of iron, is extremely unstable chemically at neutral and high pH. Chemically, it's going to undergo reactions very, very rapidly that oxidize it, not requiring any sort of biological intervention. It's just going to happen spontaneously. And because of that, because of just the chemical nature of that iron, microorganisms are not very competitive, are not able to utilize much of that iron before it's just abiotically transformed. So under neutral to high pH, iron is oxidizing, but a large portion of that oxidation won't be microbial. It's completely different at low pH, where you don't have the kind of conditions that lead to spontaneous oxidation of iron, and that's where you see microbes really ruling. That's where lithoautotrophic bacteria are very much engaged in oxidation of iron, and they're going to be responsible for the bulk of it. So, and in some cases, these organisms can tolerate extremely low pH, and we're going to look at that in a minute, just in a couple of slides. Okay, two important things that happen when iron is either reduced or oxidized, and this will completely make sense to you because we've already talked about reduced iron is very soluble and oxidized iron is very insoluble. So here's a picture of before and after some iron. It was reduced. This is oxidized form of iron, and you can see all these little granules of iron that are insoluble, but when they are reduced, you can see everything sloughs off and dissolves and goes somewhere else. And think of this as a mineral that's undergoing weathering, and how important microbes can be in essentially releasing some of these minerals that would have been precipitated out before, but then they can actually lead to their going into forms that then cause them to be moved, transported away. In contrast, oxidation often forms precipitates. Here's a picture of an iron oxidizing bacteria with all these little granules of iron now that have fallen out around it and even are coating it. And in fact, in these pictures here show these precipitated iron around this little flock of bacteria. And so they can end up coating themselves and actually committing suicide, entombing themselves in this precipitate of iron. And here's a picture where that iron has precipitated, but also with organic matter, forming these blobs of oxidized iron, oxyhydroxides here. So these processes can really lead to big changes in the environment. Acid mine drainage. Check out this article that talks a lot about the organisms that are involved in acid mine drainage. And also this article here is a really interesting metagenomic study, one of the first big environmental metagenomic studies where they essentially sequence all the organisms that were present in a situation where there was acid mine drainage. So what happens with acid mine drainage is that when an area is mined and there's going to be tailings left over, the material that isn't concentrated enough in metals to go after it, and these are going to be pyrite tailings that are piled up and left to sit there. Here's this mine site. There are these piles of tailings or an impoundment with tailings. Blow that up, and you can see that these piles of tailing are going to be exposed to the elements, to rainfall and evaporation and water moving through them in different ways. And then if we blow that up, these tailings are filled with these sulfides. Remember just a few slides before we were looking at sulfides form all these different kinds of minerals. And these sulfides are what are going to be acted upon, and that's a lot of that is with pyrite is iron sulfides with this formula. So when all that water comes in, and then there's obviously going to be oxygen coming in as well, this organism, this Thiobacillus feroxidans, which is look at oxidizing iron, which is both an acidophile and an autotroph, and can live at these phenomenally low pHs, like even pH as low as 0. They are going to be oxidizing this iron. And one of the byproducts of that is sulfuric acid, because this is also going to oxidize. And these reactions can occur without microbes, but they're super accelerated in the presence of microorganisms. So what's going to happen then is you're going to have this sudden liberation of all these metals that were present in these sulfides, and that includes iron, zinc, copper, lead, arsenic. It could be all kinds of things that are leached then from the drainage sites and often leach into these streams. And here's a picture of a stream that has that's filled with this iron that's been liberated from these tailings piles and has gone downstream. But then at some point, left as a reduced form, but they start to precipitate out into these colloidal material, this organic rubbery material like this picture, right? And fall out of solution and start to coat the stream and essentially just drown out any sort of life forms there. Can become suffocated, and this is called a yellow boy. I guess is the name for that phenomenon. And then the other thing is these streams are extremely acidic and can support virtually no higher life forms, but mainly be colonized by this crazy organism that can handle such low pH. Okay, that's the story of iron. So now let's talk about manganese. So manganese is similar to iron in having these two main oxidation states, and organisms can use it as a donor and acceptor. This phenomenon called desert varnish is a coating on rocks which has a reddish layer of clay, then with manganese and iron oxides that are, especially the iron is causing, I'm sorry, the manganese is causing this blackish color. So you can see this broken rock revealing this coating. So this coating is of black manganese oxide that is produced in part by biofilms of bacteria that are living on the rocks and oxidizing that manganese. Manganese nodules are another geologic formation that's made up of manganese salts and oxides that are also precipitated by bacteria, often at the bottoms of oceans, but then you see them in soils sometimes. These perfect little nodules, which if you take a cross section of it, you can see these like tree rings of deposition of manganese over time, where you have your initial microbial agents that then get coated by the manganese, sort of end up shutting down inside, but I think some of them are on the surface or coming from outside and continually are oxidizing manganese and getting coated by the manganese, and then a new group is oxidizing and creating these layers that you would find inside these nodules. And the other place where manganese is an issue, and iron as well, is clogging. And this happens a lot in pipes, like in irrigation systems. This is shows you a bacillus spore that has all this manganese precipitated around the outside of the spore. And it can be organisms like this, and not just bacillus, but others as well. If they do oxidize the manganese, they can just create this essential layers of precipitation that end up just filling the pipe up until water can't drain in them anymore. Okay, mineral weathering is a consequence of microbes interacting with elements that are in these minerals, and through the issues there are dissolution of minerals and corrosion, which is a term that we use when types of materials that we as humans are wanting to use for certain purposes, they ultimately will also be colonized by microorganisms with negative impacts on what we want to use these materials for. So when a new surface, be it basalt or be it a ship hull, goes into an environment that has some water in it. It could be in soil, it could be in groundwater, it could be in an ocean. You immediately get colonization of the surface by bacteria that often attach by stalks. So even a clean surface will then start to be coated with these organisms, and also with a layer of organic material as well. And here's a little bit later in this succession, where this material's been longer in the, for example, the water, where you start to get this rough surface. It's made up of biofilm of organisms. It'd be extracellular polymers in there and other bacteria. And then over time, you can start to have the surface be essentially picked at or mined by the organisms for any kind of element in there that they may be able to use as a nutrient. And if you look at this at a small scale, you actually, it's quite interesting. You see like a little mini electron tower in it, where you have, once this biofilm has matured and gotten thick enough, if you were to look in it, you would see that at the outside of the biofilm, you have oxygen being used as an electron acceptor, but it's not going in very far because it's used up at the surface. And then you're going to have, I mean, nitrate and iron, if they're present, would be used. But as you go closer to the surface, underneath this thick biofilm, that's where you're going to see the sulfur used as an electron acceptor. And that sulfur is going to be reduced when it is an acceptor to H2S. And then that H2S, because it likes to form new compounds with things like iron or any sort of cation around, is going to start to pit the surface by plucking out cations, for example, iron, and creating ferric sulfide as well. So this can lead to then corrosion of material that is made possible by this special environment that is anaerobic, that involves sulfate being used as an electron acceptor, that allows organisms to then go and wreak havoc on these minerals. And so corrosion with man-made materials, but this would be like a kind of mineral weathering with natural material. So the mechanisms involved are, oh, and I didn't talk about the fermenters, because the fermenters also are producing electron donors for that sulfate, right? The carbon that's around in the surroundings will be fermented in this middle layer and creating the donors that the sulfate reducers can use. So mechanisms are formation of acidity, the lowing of the redox in this area, and then the H2S pulling the iron from the surface. And here's some examples of some corrosion. I'm sure you've all seen corrosion in your life. Of, these are the kinds of consequences that can come from microbes going at it for quite a while through the process that we just talked about. And here's a recently corroded material with some iron in it, and then over time, well, this is actually a close-up of it showing this essentially fracturing of this, what was otherwise a continuous metal material, and start to break it apart and crack off bits of it. Okay, mercury, let's talk about mercury now. Issues with mercury is methylation of mercury increases exposure and toxicity for sensitive species, by the way, including humans. And I just want to put in parentheses that this kind of process also applies to arsenic, but we're not going to talk about that in detail. But methylation is a microbial process. But let's back up and know that one of the legacies of the gold rush in California was the central stockpiling and then release of mercury all throughout the state, because mercury was used to help in mining the gold. So wherever there was this kind of modification of the environment for gold, there's a lot of mercury. And this is a figure showing mercury that's found in microbes in these different environments, where the higher concentration is found of mercury in the streams that had the most gravel mined, that had the most exposure to mining activities, and a pretty direct relationship between that, how much mercury is going to be there with it in microbes as an indicator, and how seriously it's been impacted by mining. And this picture just shows you what can happen then, and we're seeing this in many places in California, that this mercury that may have been brought to a location where they were mining for gold, or mercury that may have been mined for its own self, becomes pulled to the surface, and then it's mobile, and it moves into freshwater water bodies, and has its own little cycling, which we'll talk about in a minute. And then some of this also goes to oceans. So we definitely have a pretty substantial mercury presence in the state in waters throughout, because of this legacy of the mining activity. So microorganisms can do a lot with mercury, and this picture shows you a mercury cycle in a freshwater lake. And things that microbes can do is oxidize and reduce mercury between the elemental +2 and +3 phase. Oxidation reduction reactions. They can also methylate mercury. That means they can put essentially a methyl group on the mercury, and that's usually starting with the +3 form. So they may be involved first in oxidation of the mercury, that then is a form to be methylated. And organisms that methylate mercury include sulfate reducers and iron reducers. We don't exactly know why they're methylating it, because it's expensive to methylate. You have to use energy to get that methyl group on the mercury. It's a conjugation reaction, just like siderophore and iron. That's a conjugation reaction. So people think maybe it is to detoxify the environment. They feel that if the methyl group makes it mobile and it gets it out of the way, and that's of advantage to the microbes, but we don't know exactly if that's true. And also, there are microbes that demethylate mercury. They might see that methyl thing hanging off of the mercury going by, and they watch it and say it looks mighty tasty. There's a carbon source, and so there's some that it can actually demethylate the mercury. So methylation is a big issue of mercury because it increases the mobility of the mercury, moving it from where it might have been behaving well, down in the sediment, not moving around very much, just staying put and not being engaged that much with organisms in the water column, particularly fish that people catch. And also it has a high propensity to be bioaccumulated into tissues of living bodies. So that's number 2. So it's really easy for it to build up really, really fast in organisms, unlike just regular mercury, non-methylated mercury. And the other thing is it's extremely toxic. So you don't need high concentrations for it to be toxic. And so this has actually resulted in a situation where a lot of fish, I think I just heard a story on the news about Seattle having a lot of mercury in its fish, but you can count on a lot of fish being contaminated with mercury that comes from these processes that I just talked about, and the movement of mercury from these tailings into freshwater reservoirs and ultimately into the ocean because of these microbial processes transforming the mercury and leading to the methylation, that it's set up this situation where now we have a lot of mercury in fish. And there's been these studies that have shown, like in the Bay Area, that almost 90% of people tested there, men, women, and children, had 90% had blood levels that were higher than what was deemed as a safe level, sometimes 7 times that. And then they've also been able to demonstrate that the mercury goes down when people stop eating the fish. So it definitely seems to be a relationship. And there's all these like public service types of posters like this one that you can find. And I know people have discussions about it. I don't eat fish, so I don't ever think about these things, but I hear about people worried about like the kind of fish they eat, and is it a bottom feeder, or how high is it on the food chain? Like, is it a fish that's bioaccumulated a lot of mercury because it's eaten smaller fish who have eaten smaller fish, et cetera, or are they more direct feeders and therefore less likely to accumulate mercury? But anyway, these are some pictures that give you ideas of that, with this little thermometer showing how much mercury these different fish have in them. But that's a real concern, and there's a direct relationship to microbes. Okay, selenium is another element that can be methylated, but in this case, it's a different story. The selenium is actually, it's beneficial, at least to humans, for selenium to be methylated, because it can decrease exposure of both wildlife and humans that might be exposed to it, and help in remediation. And there's some really good sites. This one here, a website, will show some really good examples of application of these remediation techniques to selenium. Look at selenium. Isn't that uncanny? What does it look just like? That's right, it looks just like sulfur. It's so much like sulfur, it isn't funny. And that's the reason, in fact, that selenium is quite toxic to some life forms, particularly to birds and wildlife. And this picture shows you this one location in California where there was, everything sort of came to a showdown about selenium. So let me develop that story in a minute. But these poor birds were exposed to so much selenium, and the body, in some cases, the enzymes can't differentiate between sulfur and selenium. So these would be like an amino acids that are supposed to have sulfur in them. When there's a lot of selenium around, they'll go ahead and incorporate the selenium into those amino acids, and that ends up making lousy enzymes or whatever it is that they're making from those amino acids, and leading to deformation or death. And so that's the issue with exposure to too much selenium. So because it looks like sulfur, but it ends up making inoperable or poorly operable enzymes. So anyway, all these different states, just like sulfur, and undergoes the same kind of transformations, and a lot of the same organisms that are involved in all these transformations for sulfur also are engaged in selenium. And then there's a bunch of different kinds of microbial processes, mobilization and transformation of selenium oxyanions, mineralization of them, and then methylation, which is shown here with this form of selenium. It is the one that's susceptible to this methylation process that can be carried out by algae or bacteria. And when the selenium is methylated, it's volatile. And if it's volatile, then it can waft into the air and take off and end up remediating the whatever is the system, the pond or whatever holding tank where there was this selenium. It's a way of getting rid of it by sending it to the air. Now, is that a good thing? Does that create a problem in the air? Well, probably not. Selenium isn't that toxic, and particularly not in the air. It would be quite dilute, and so I don't think there's much concern about exposure to that. And in fact, if it would just blow over from the west side of the valley of California to the east side, it would actually bring much needed selenium to the cattle over there on the east side, and would solve a nutrient deficiency problem over there. Isn't that weird? Like in the state of California, you can have this discrepancy of over too much selenium in one side of the Central Valley and deficient conditions of selenium on the other side. There's some great review articles here you can check out. And I mean, where everything came to, that's nice, it came to a head was in Kesterson. I don't know if a lot of you have heard about this, but Kesterson is a location for a lot of really big scale evaporation ponds in the past, where irrigation water, after it's run off of fields, would be collected there to just evaporate the water. Then the days when we didn't have as many concerns about water, to be able to just get rid of it, right? Because there was too much water. And then, more water would be pumped there and it would evaporate. And this water is just naturally high, the irrigation water, with selenium that comes from the coastal ranges. So because there was irrigation and a lot of sort of moving water over these systems, a lot of selenium got essentially mobilized from the natural deposits it was hanging out in and moving into irrigation water. So as the water evaporated and kept filling, the selenium concentrations got quite high. Unfortunately, those ponds that had the selenium water in them are very attractive to birds, so a lot of birds ended up landing there and nesting there. And then that's when people started to see these deformities, and really, really extreme deformities in the birds that were using those ponds and having offspring. So there was a lot of really interesting research for a while, including some microbiology, to see if they could use these biological processes and add, whatever is needed to the system to be able to naturally remediate these sites. So they played around with all different kinds of organic compounds that could be used as sources of the methyl group, and they also studied which kind of microorganisms are involved and what could they do to make conditions more conducive for them so that then they would methylate a whole lot of mercury. A lot of research was done on this. I think they found that the rates just were not high enough to be able to make much of a dent in these big ponds, and it was pretty much abandoned as a tier 1 remediation strategy. And I think what they've ended up doing is just paving these areas for the most part, because they didn't really figure out a way to remediate them of the selenium. Okay, that's the end of the story of selenium. So there's just some supplemental information. Here's the whole genome for the mine site. The whole genome for that acid mine drainage site that I talked about before, where they were able to identify all the enzymes that were present in just a small group of organisms living there. So it's a kind of cool paper. Check it out. And then here's some just pictures of all these interesting kinds of sculptures that are produced by microorganisms, and you can read about it here at small scales. The way they modify the environment through these kinds of reactions that we talked about in this lecture were the driving force of those. So I end here, and that is the last lecture in Soil Science 111. Okay, thanks very much.